Okay, so today we're going to be talking about plasmids. Plasmids are very small circular pieces of DNA. They're very easy to manipulate, meaning so that they're going to be used in genetic engineering and stuff like that. And they are mainly found in prokaryotic cells, such as bacteria. So when we're actually looking at the different parts of a plasmid, we can see that we have the origin of replication. The origin of replication is going to technically be anywhere on the plasmid, but that's going to be where DNA replication is going to start. We have the promoter. So the promoter is where transcription will start on the inserted gene. So similar to DNA replication and DNA transcription and translation, stuff like that, the promoter is going to be where transcription will start. A lot of plasmids actually contain antibiotic resistant genes. Usually this gene is tetracycline, ampicillin, or kanamycin. These vary, but those are the main ones that you'll find. You also have your selectable marker. The selectable marker is a little bit harder to explain, but the selectable marker is a gene that will allow scientists and researchers to actually see if a cell has taken up a plasmid. If you were to mix bacteria and plasmids together, the selectable marker would actually be able to determine through plating, etc., to see if a bacteria has actually taken up a plasmid. We also have inserted genes, so this is literally the gene that is going to get inserted into the plasmid. It's important to know that once this gene gets added and once it then becomes a vehicle for genetic engineering, then it is considered a vector. Only until this gene gets inserted would this still be a plasmid, if that makes any sense. So why do we actually want to use plasmids, and why do we want to use vectors, and what benefit do they actually have towards research and through genetic engineering? Naturally, plasmids offer advantages to bacteria, like I said, just in terms of like antibiotic resistant genes. The reason we care about them though is so that we can make vectors that will actually help with gene transferring, gene cloning, etc. So there are different types of plasmids. There are conjugative plasmids, which transfer only through conjugation. I have not gotten into transformation or conjugation or anything like that yet, which I can do in a different video. But essentially, conjugation is just, it's a part of horizontal gene transfer. It doesn't have to just be towards antibiotics, but any plasmids that have a resistance gene to it, more commonly it would be antibiotic. They're considered R plasmids or R factor plasmids. And then there are virulence plasmids. Virulence plasmids are plasmids that carry genes that would make them more pathogenic. So they could have genes that code for better adhesion. They could have genes that code for enzymes that break down your body's immune defenses, stuff like that. So anything that will make a plasmid or whatever bacteria is hosting the plasmid, it would make it more pathogenic. So like I said earlier, plasmids are just naturally occurring circular pieces of DNA in prokaryotic cells. The reason we care about them though is so we can use them as vectors, and then vectors are anything that we can use in genetic engineering, such as gene cloning, gene expression, gene transfer.